Hello, people of science. We are going to talk about cell transport. Hopefully, we're going to answer those questions of how do cells control what enters and leaves the cell, and why is this actually even important? Why are we learning about this? So here's kind of a diagram, obviously a rough schematic of a cell, but you can see things are moving in and out. Whether it's glucose or amino acids, a cell needs to regulate that. That's what this is all about. So, you don't need to write this down. Hopefully you should know this by now. The cell membrane, we've talked about it a lot. It is semi-permeable. And remember, that means some stuff can enter and exit and other stuff can't. This is like the security guard or like the bouncer of the cell. That, the person who says, like, you're not allowed in here. And I don't go to a lot of places with bouncers. But I wouldn't mess with that guy. I do what he says, because he's the one who obviously is in charge of determining what can go in and out of a cell. So, why is that even important? Well, when you get too many things entering or leaving a cell, the cell, and you by extension, will die. You don't need to write this down again. This is just why we're learning about this stuff. This is a red blood cell. This is a normal red blood cell. It's happy, it's healthy. It looks like a kind of squished donut with a donut hole in the middle there. It's not hollow. It's just kind of squished down. That's normal, that's happy, that's good. Here's one that's about to get all explodey and stuff. It's supposed to have this cute little dimple there. This, not good. If you were to go to the doctor and be medically treated for dehydration, they are not gonna, hook up water to an IV and plug it in your arm, because that would kill you. It would kill your red blood cells, making them go all explodey, and by extension, it would kill you. So instead, what they're going to do is they're going to put a drip in you of 0.9% saline, which is a very, very weak saltwater solution. That is something that will not kill you, which is why you generally want a doctor that's not trying to kill you. So maybe it's a good thing that they're putting the saline in you. Well, let's talk about why. There are types of cell transport. This is where I want you to start writing. We have two big categories, passive transport and active transport. And as you can see, in passive transport, no energy is required. Active transport, you need energy in the form of ATP, adenosine triphosphate. We'll talk about that way more later. But we have three types of passive transport and 5 different types of active transport. Well, let's go through them. So first, we're going to focus on passive transport. Everything here in blue does not require energy, no ATP. This just happens on its own. So that first one we have is diffusion. Now this is the movement of molecules without energy. These are things that move from an area of high concentration to an area of low concentration. So right here I have all these molecules, these red little dots here, that over time are gonna move from where it's really concentrated to not very much. And you don't need energy. Again, no energy is required on their own. They're going to just diffuse out and reach equilibrium. They're going to spread out there. So it goes from looking like this to more like that, where it's evenly spread out. We call that equilibrium. So that's diffusion. And again, no energy. Second type we have is called facilitated diffusion. That comes from the actually Latin and Spanish root word fácil, meaning easy. This is just something that actually helps things diffuse. Again, does not use energy. This is where things move from a high to low concentration, but they can't get through normally. Like in the other video, the dog that was in the cage, they can't just go through the cage on their own. They have a special doorway. So facilitated diffusion, you're still going high to low, but through a special doorway. And these doorways are called channels or carrier proteins. Here you can see another one. Too big to fit through the membrane on their own, go through a special protein door. Now we have osmosis, not Osmosis Jones, that's just a show and movie, whatever. Anyways, this is the diffusion of water across a semi-permeable membrane. We'll do a lot more about that, but this is where you see cells that can either swell, like that red blood cell we saw, or ones that can actually shrink. We'll talk about that one later. Water will enter or leave a cell, going always high to low. Remember, no energy, it's going high to low. So here's a cell in a hypertonic environment, it is losing water. Here's a cell in a hypotonic environment, it is gaining water. In this case right here, it's just swelling, it's about ready to explode. That yellow represents salt there. And the isotonic solution where something is happy. Now remember, water cannot actually directly cross the membrane, so it uses special doorways called aquaporins. That's one of your vocab terms. So here you can see just an aquaporin, water passes through that channel. So again, all of these passive transport, No energy required for diffusion, facilitated diffusion, or osmosis. Let's talk about active transport. These are things that need energy in the form of ATP. They are endocytosis, pinocytosis, phagocytosis, exocytosis, and bulk transport. All of these have to have energy, won't happen on its own. Well, let's take a look at them. First off, endocytosis. This is just a generic term where a cell brings things in. Like here you can see endocytosis, that cell is bringing in a whole bunch of stuff. Endo just meaning entering or in. So that's endocytosis, just a general term. Next one here, we have pinocytosis. And I know what you're thinking. It sounds like this. If you like pina coladas, get caught in the rain. Okay, that's enough of that. But that's like pinocytosis. It's, you know, if you like pinocytosis. All right, I'm stop. I will never sing again. But they are related because it has to do with water. A cell is taking in liquid. It is drinking. So here you can see liquids and other small molecules, but mostly going into the cell there, pinocytosis. Next up, phagocytosis. This is a general term for when a cell is taking in food. It's taking in eating, like phagocytes or esophagus. All those come from that same root word. Here we can see we got something too big to just be absorbed. So phagocytosis is that cell gobbling it up and eating it. And exocytosis, those other ones you saw were in. Exo obviously means out, or out. A cell is getting rid of or removing waste. A cell is pooping. You're looking at a cell pooping. Bet you didn't think I'd make you look at anything pooping this year. Too bad. You're looking at a cell pooping. Poop. And finally, we have bulk transport. When you're buying something in bulk, like buying toilet paper in bulk, because there's no respectable amount to buy. You're either buying 500 or one at a time. Anyways, you're not moving stuff one at a time. You're moving large quantities in or out at once. And now that I say that, it sounds like I'm referencing poop again. Sorry. And that even kind of looks like poop. This recording took a turn for, well, the poop, I guess. Anyways, this is a large molecule entering the cell. Bulk transport, not one molecule at a time. Large chunks of stuff. So, just to recap, we've got endocytosis, pinocytosis, and phagocytosis. Those are things that something is coming in. Exocytosis, something is going out. And bulk transport, large things are moving either in or out. So, these all right here are active transport. These all need energy. Endo, pino, phago, and exo, and bulk transport all require energy, whereas the passive ones that we saw before, diffusion, facilitated diffusion, and osmosis, none of them do. All right, that's it. I'll just play you out now.